Hi, everyone. This is Dr. Sharon Thomas, and I'm going to be proctoring the Designer Genes competition at both the regional and state levels. And so I wanted to spend a little time with you today to describe the event and what can you expect on the exam. So um, the event description is stated right there, and you can read that on your own. It will be a team of up to two, and the approximate time of the exam, which will be delivered through Google Classroom, will be about 50 minutes. You may have a cheat sheet with you, which is an 8.5 by 11 sheet of paper, which can have any kind of information on both sides that you wish to bring into the exam. And the competition is going to be not run as stations this year because of COVID restrictions, but it will be a multiple choice, short answer, and free response test given through Google Classroom. So let's just talk about the topics. We're going to be running, any of these topics will be fair game for the exam. So mono, di, and trihybrid crosses, those can include, for the di and trihybrid crosses in particular, linked genes, unlinked genes, so make sure that you're familiar with those. Dominant and recessive alleles, I think that's pretty clear what that would mean. So whether you see the presence or absence of a particular phenotype in future generations. Human sex determination is quite a broad topic. Just make sure you know the Y chromosome is the sex determining chromosome in humans, and a little bit about the SRY gene. So make sure you're familiar with that. Make sure you are familiar with transcription and translation. How do you get from gene to protein? Mitosis and meiosis and gamete formation. So make sure you know the differences and the similarities between mitosis and meiosis and how gametes are formed. For human karyotype analysis, make sure that you're pretty well aware of how we get abnormal chromosomal configurations in human karyotypes. That would include things like non-disjunction, translocations, those kinds of types of mutations that happen to give you an abnormal karyotype. Also, make sure you know what can and cannot be seen with a human karyotype. So for instance, we can see the number of chromosomes and whether any chromosomes are missing or broken, or there are extra chromosomes, but you really can't see alleles or phenotypes, and you can't determine phenotypes based on karyotypes. Make sure you're familiar with prokaryote and eukaryote promoters and the components of a gene. So that would be exons and introns and enhancers and all of those things that make up a gene in both prokaryotes and eukaryotes. Be able to interpret or draw a pedigree, and make sure that you know the difference between multiple alleles, and that would include allelic series. So most genes have many alleles associated with them, so make sure you know what alleles are and what it means for an allele to be dominant or recessive. Sex-linked traits are often passed along the X chromosome, so make sure you're familiar with what a sex-linked pedigree might look like, or what are some common sex-linked traits out there. The difference between codominance and incomplete dominance, just make sure you know the difference between those two, and you can determine which is which given an example. Make sure you understand how genes are expressed and the role of enzymes, and some of those enzymes can be like RNA polymerase. Make sure you understand epistasis and multifactorial traits. So epistasis, an example of that would be albinism. And multifactorial traits would be genes or traits that are controlled by multiple genes, like height. Make sure that you understand the difference between hypomorph, and null alleles. Those are the molecular consequences of mutations that can lead to recessive and dominant traits. DNA structure, of course, DNA is double helix, and you should be able to label the parts of a DNA structure or draw one. And also, what's new this year is repair. So make sure that you know the five major mechanisms for DNA repair after replication or when mutations are induced. Phylogenetics, make sure that you're able to interpret or draw a phylogenetic tree given specific amino acid percentage differences, things of that nature. And DNA fingerprinting is pretty broad, and it's changed a lot over the years. So for DNA fingerprinting, make sure that you can interpret a gel that has DNA fingerprinting results on it. That would include even knowing how gel electrophoresis works. And RFLP is restriction fragment length polymorphism. So you need to be able to identify how restriction enzymes work and how those restriction enzymes can help you do DNA fingerprinting. Polymerase chain reaction is a way to amplify small pieces of DNA. Make sure you know the steps of PCR and really the applications of PCR. So what is PCR used for? DNA microarrays are another biotechnology tool, and these can be used for a number of different things. So make sure that you understand how they can be used to measure expression levels, or they can also be used for DNA genotyping. So make sure that you get some examples of DNA microarrays, look at them and see if you can interpret the results. We also are going to could be talking about gene therapy and CRISPR-Cas9 technology. So CRISPR, of course, is the greatest and neatest tool to modify genomes of living things. And so make sure you understand the mechanism of CRISPR and some of the drawbacks that can be involved with CRISPR technology. Make sure you take a look at the Sanger and next generation sequencing. So why would we want to sequence genomes, and what does that sequencing tell us? And so what can sequence DNA give? What kind of information can we get from sequenced DNA? Plasmid cloning, selection, isolation, and random versus targeted mutagenesis. That is a lot in that box. So this also goes with restriction enzymes. So plasmid cloning usually entails taking a plasmid, inserting a foreign piece of DNA within a plasmid, and then that plasmid then can be used to make a GMO, or it can be used to express a protein in a bacteria or some other lower organism. And you can also then take that plasmid and change DNA components with it, and that would be random versus targeted. So if you know which base you want to change and you have the ability to change that, that would be targeted. If you're just going to allow the plasmid to replicate and hopefully get some random mutants out of that plasmid, and you have no control where those mutations occur, that would be random. And then, of course, always make sure that you know mitochondrial DNA and chloroplast DNA and the kind of mutations that are associated with mitochondrial DNA and how that organelle, in particular the mitochondria, how it's passed among generations. So there are some sample questions down here which are pretty good, and they're talking about mutations and sequences and going from DNA to RNA to protein. And then, of course, there's some electrophoresis sample questions here. We're going to be scoring the highest number of correct solutions will determine the winner. And then we will also have some tiebreakers at the end. Please try all the tiebreakers, because those tiebreakers have been used in the past in order to determine a winner. I hope all this is helpful. And if you have any questions, again, my name is Sharon Thomas, and my email is sthomas@ortn.edu. And if you'd like to shoot me an email with a little bit of clarification, just let me know. Have a good day.